Is there a crime gene? Many people in the field agree that there is no crime gene as such. Genetics may account for, say, half a person's aggressive behavior, but that 50% comprises hundreds or thousands of genes that express themselves differently depending on the environment. When these switches that can be associated with violent and aggressive behavior are present, when a child is not exposed to environmental risk factors such as abuse, poor attachments, and peer delinquency, they appear, based on the research, to remain latent. However, where multiple risk factors exist, genes become activated and account for, according to the research, 80% of violent behavior. Walsh and Beaver, 2009. Other recent neuropsychological research also focuses on traits that are typically associated with criminal behavior, including self-control, callousness, and a lack of empathy. Like other personality traits, these are believed to have environmental and genetic components. Although the degree of heredity is debated, but a predisposition is not destiny, and gene criminality research continues to prove so. Epigenetics. According to a relatively new field in behavioral genetics, epigenetics, interactions with the environment can cause changes in gene function that occur in the absence of changes in DNA sequencing and can be inherited by future generations. DNA has been traditionally viewed as the blueprint for everything concerned with human life, which is inherited and fixed and therefore predetermines the physical, mental, and behavioral fate of individuals. Recently, this view has been challenged from various disciplines, including behavioral, genetics, psychiatry, and social sciences, which postulates that genes are in constant interaction with the environment, and although the genetic code is not changed in the process, the way the blueprint is interpreted is adjusted. Epigenetics is the study of heritable changes in gene function caused by mechanisms other than changes in the underlying DNA sequence, hence allowing for different interpretations of a fixed template and result in different readouts, which is dependent upon the variable conditions under which this template is interrogated. DNA is wrapped around proteins called histones, both which are covered with chemical tags known as epigenomes. The epigenomes shape the physical structure of the genome and essentially wraps inactive genes and relaxes active genes, thereby making them accessible in expression. Therefore, the DNA code remains fixed for life. The epigenome is flexible, and epigenetics tag react to signals from the environment, such as diet and stress. In this context, the environment is referred to in a broad sense and includes hormonal, social, nutritional, toxicological exposure during prenatal, postnatal, adolescent, and adult life stages, according to Champagne 2010. Many physical and mental behavioral characteristics have been attributed to, or in part, to changes in epigenome, including antisocial behavior. According to Moffat 2005, the gene-environment interplay, gene times F, has a far larger impact on psychopathology than its individual components, and especially involve genes whose effects are conditional on environmental risks, such as malnutrition and abuse. As such, genetic and environmental risks are often concentrated in the same population segment. The most potent risk for pathological behavior is presented. Recent research has eliminated the potential effects that environment has on gene capacity to influence phenotypes, such as sensation seeking, overactivity, fearlessness, low self-control, negative emotionality, callous and unemotional style, weak verbalization, poor memory, executive dysfunction, frontal lobe hyperarousal, serotonergic dysfunction, and testosterone imbalance, page 458, which all characteristics of antisocial behaviors associated with criminal behavior. monoamine oxidase. Monoamine oxidase is an enzyme that is encoded by MAOA gene in humans and is preferentially diminuates non-epinephrine, epinephrine, serotonin, and dopamine, the process by which these neurotransmitters are degraded. Elevated levels of MAO-A have been associated with major depressive disorder and sleep disturbances. A low activity MAO-A gene has been associated with the so-called warrior gene. It is linked to aggressive and antisocial behavior, in particular reactive situations when provoked. If this genetic variant is present, it seems to be switched on by substantial child maltreatment, according to Gottschalk and Ellis 2010. The tainted history of using biology to explain criminal behavior has pushed criminologists to reject or ignore genetics and concentrate on social causes, poverty, addictions, guns, among others. Now that human genome has been sequenced and scientists are studying genetics of areas as varied as alcoholism and party affiliation, criminologists are cautiously returning to the subject, exploring how genes might heighten the risk of committing a crime. According to Ray in 2002, one reason why we have been so unsuccessful in preventing adult crime is because interventions today have systematically ignored the biological side of biosocial equation that produces crime, in combination with environment, genetic factors of humans, eminent factors determining psychopathology and behavior.